Start by introducing myself. I'm Dennis Eastburn. I'm Chief Scientific Officer and a co-founder of Mission Bio. And thanks to all of you for coming to our Spotlight Theater today. We have two talks from fantastic collaborators, Catherine Smith from UCSF and Aaron Vinney from Memorial Sloan Kettering. And they're going to talk to you today about work they've been doing using our single-cell DNA sequencing technology to better understand clonal architecture within tumor populations and how that better understanding can ultimately lead to different clinical decisions. But before you hear those two headliners, I'm going to tell you a little bit about the technology that's enabled the research they'll be presenting. So it's now fairly commonly appreciated that many cancers and tumor populations are quite genetically diverse. And it's often rare or heterogeneous populations within tumor populations that can drive important aspects of tumor biology and progression of the disease. And a specific example of this is sort of illustrated on this slide. At the time of diagnosis, if you have a genetically heterogeneous population, you can imagine that treating with a therapy such as chemotherapy, what you're doing with that therapy is basically selecting for certain subclones within the population that ultimately have a fitness advantage in response to that treatment. And of course, those clones that have the fitness advantage can ultimately end up relapsing, and then you treat with another therapy, and you can go through multiple rounds of this, and ultimately, the prognosis is often not good for patients that kind of go through these cycles. So our sort of mission has been to develop a technology that can better, sort of parse apart the totality of heterogeneity that exists within tumor populations, and analyze that throughout the course of the disease, so that better therapies such as precision medicines can be selected to not just, target one population or two clones within a sample, but really to better target all the tumor populations that exist, and to sort of break this cycle of treatment and relapse. So basically, to accomplish this, we've invented and developed a technology that we've now commercialized and launched. It's our Tapestri Precision Genomics platform. And it really enables broad applications across oncology. And I'll just go through a couple key features of this technology. Fundamentally, what it does is it targets up to 300 different genomic loci for very accurate SNP and indel calling. And not only can we target up to 300 different positions throughout the genome, but we do that across 10,000 individual cells per sample. And so it's really that high level of throughput that enables a very high sensitivity ability to detect rare clonal populations important for tumor progression. And we've purposefully made a workflow, the Tapestri Genomics platform workflow, very simple so that it's accessible to both researchers and clinicians. And as part of making that workflow simple and accessible, we've paid particular attention to the downstream bioinformatics and data analysis portions of the platform. So they're actually quite intuitive to use and provided free of charge as part of the solution. The platform is compatible with both hematologic samples, as well as now we're making it compatible with solid tumor tissue. And together with that, we offer not only an array of fixed panels that target specific genes relevant to both solid tumor applications and hematology applications, but we also have the ability to allow researchers to take genomic regions of interest to their particular application and actually put that onto the genomics platform for their particular research. So as I mentioned, the workflow is quite simple. It is designed to fit seamlessly into existing NGS workflows that labs are already performing. At a high level, you take a cell suspension or a cell prep of interest, and you load that onto our Tapestri instrument, which runs disposable microfluidic cartridges. And if you haven't seen the instrument, it's quite small. It's about a cubic foot in size, and it's actually on display at our booth. And so once the instrument and the cartridge perform what we refer to as our two-step workflow, you're ready to proceed with a relatively simple downstream library prep followed by next-gen sequencing. And then, of course, you can utilize the data visualization and analysis package that we've developed. So I want to spend just a moment talking about the microfluidics that run on our disposable cartridge, because it's those droplet microfluidics are our two-step workflow that is really unique to Mission Bio, and it's what differentiates our technology from other droplet workflows you may have heard about and that are commercially available at the moment. We first take cells of interest and we flow them together with a proprietary lysis reagent. A proprietary lysis reagent containing a protease. And so once droplets have formed, those cell suspensions or cells and the protease buffer are allowed to mix, and the protease not only lyses the cells, but it goes in and actually digests nuclear proteins such as histones that would otherwise sequester genomic DNA and hinder downstream molecular biology applications. Of course, the use of a protease necessitates that we turn its activity off once it's performed its function. And at that point, you have a population of drops with prepared genomic DNA lysate that are ready to go into the second step of our workflow that's also performed on the same microfluidic cartridge. And there, those genomic DNA containing droplets get combined with unique cell-identifying barcodes, PCR reagents, as well as targeted gene-specific primers for the loci of interest. What's going on with the mic there? Once those drops are basically formed, you have your complete PCR reaction in your droplets, and you're ready to put those droplets on a standard laboratory thermocycler. There, the barcodes or cell-identifying barcodes get incorporated into the amplicons produced from your targeted genomic loci. And so you might ask why we've gone to the trouble of generating this two-step workflow. And it really stems from very early data we obtained during the development of our technology, showing a real advantage to using a protease. And I'll sort of just, one example of that data is shown to the right there, and I'll sort of just cut to the chase and say that we have ways of running our workflow both with and without a protease. And when we use a protease or incorporate a protease in the workflow, we consistently see much better amplification at our targeted loci, and the net result being that we have much deeper sequencing coverage at the genomic loci of interest. And that enables really, fundamentally our very accurate SNP and indel calling, at disease-relevant loci throughout the genome. So the technology is capable, as I mentioned, of very high sensitivity detection of subclones. And this is just a development experiment where we showed that we can take known cell types and, running, cell samples at, 8,000 to 9,000 cell throughputs. We can spike in concentrations of known cells and detect them down to a 0.1% sensitivity. And so it's that capability, that high sensitivity variant detection capability, combined with the ability that's unique to single cell analysis to unambiguously show co-occurrence of variants within a subclonal population that, again, is really unique to this platform. And it's really something you just cannot get through bulk next-generation sequencing. And I think the talks you'll see very shortly from our collaborators will really demonstrate how both that sensitivity and ability to show co-occurrence is really impactful when it comes to understanding tumor biology. So I'll just briefly mention our software solution. Again, we recognize that there's challenges in analyzing this type of data, so we've really worked hard to develop a user-friendly and intuitive solution that's cloud-based and provided free of charge to our users as part of our solution. We have basically two steps to this solution. You first take your next-gen sequencing files and you upload them and do variant calling in our Tapestri pipeline. Once that's performed, you can take those variant calls and actually use our Tapestri Insights package to more carefully analyze your data, look at specific clones and the variants within those clones, and even compare clones across multiple samples, either longitudinally or between patients. We initially launched our platform last year with a focus on AML, and we introduced a 19-gene focused AML panel, and that's actually what was used in the research that will be presented in just a little bit. But we're very excited to announce that we've now developed a 47-gene myeloid panel that we believe will greatly expand our applications in the hematology area. Oops. And then lastly, we also are now introducing our cancer hotspot panel, which targets 50 genes that are relevant to the acquisition or progression of many different indications, cancer indications. And so it covers a lot of the common oncogenes and tumor suppressors associated with different types of solid tumors. And in conjunction with the development and launch of this panel, we've actually developed methods to disaggregate and analyze solid tumor tissue through nuclear isolation. And so the nuclear isolation method we've developed is quite simple. It takes less than an hour and requires no extra hardware. And we've actually gone ahead and shown that you can use this nuclear isolation method to isolate nuclei from breast tissue, lung tissue, colorectal tissue, as well as prostate tissue. And we get very similar performance from those nuclear preps as we do with our standard previously validated cell lines or bone marrow aspirates or PBMCs. So we're very excited about both the introduction of this panel as well as this method to actually isolate solid tumor, basically single cells. So with that, I'll just wrap up by saying, we've developed and launched an industry-first ability to very accurately call SNPs and indels at up to 300 different positions. And we do that at extremely high throughput, 10,000 cells per run, which enables exquisite sensitivity. And we're providing both multiple fixed content panels, as well as the ability for you, the researcher, to develop your own custom content for targeted sequencing on Tapestri. And we spent quite a bit of time developing meaningful data analysis and software tools so that you can make sense of the data that you generate with the platform. So with that, I will turn it over to Kathy Smith.